Hello, biologists. Today we're going to talk about DNA replication. And the first thing we're going to discuss is the structure of DNA. We know that DNA is a type of nucleic acid, and then it's made of monomers called nucleotides. A single nucleotide is composed of a phosphate group, a sugar, a deoxyribose sugar, and a nucleic acid. So those are the three components of the nucleotide. We have 4 varieties of nucleotides that we find in DNA, but we have 5 total because when we're talking about RNA, which we're not really going to today, but when we do, we know that there is one more, and it is uracil. So we don't want to forget that uracil is a nucleic acid, and it is involved in our cells. But for DNA, we have 4 types of nucleotides. Those 4 nucleotides do complementary base pairing, meaning that A always binds with T, C always binds with G, and that continues forever and always, which helps us because if we had one single side of the DNA molecule, we could tell what its complementary side would be just by looking at the pattern of nucleotides in the side that we have. The nucleotides are bound together in the center of DNA with hydrogen bonds. That's an arrow. So here we have hydrogen bonds holding together adenosine and thymine and cytosine and guanine. There's a difference whether or not it has 2 or 3 hydrogen bonds. And those hydrogen bonds are essentially like Velcro. So they're really easy to pull apart and stick back together, pull apart and stick back together, which is great because we're going to be pulling apart our DNA and separating the 2 sides of our double helix anytime that we want to make a piece of mRNA or anytime that we're going to replicate our cell. So we're doing that constantly. We want to be able to pull those apart really quickly. And that's why it's like Velcro. The other sides, though, here, where you have 2 nucleotides of one side of your DNA stuck together, that is a phosphodiester bond. That's a type of covalent bond. It's really strong. So when you pull on your DNA halves, you can split right down the center, but you're not going to pull your individual nucleotides apart along your sugar phosphate backbone. Those are going to stay together because those bonds are quite tough. The hydrogen bonds themselves are really easy to separate. You can just separate them by heating up DNA. So if you get it warm enough, they just kind of fall apart. When we look at our single nucleotide, at the top of the nucleotide, you're going to have an oxygen. I always think this kind of looks like a little person with a rotund body and 2 little arms. So that helps me orient myself as I look for the little person. But the oxygen is at the top. And when we identify a nucleotide, we count the carbons going clockwise from oxygen. So we go here and around from the oxygen. On the corners, anywhere where you see a junction between 2 black lines, the assumption is there's a carbon right there. That's conventional based on how we draw chemical formulas. So if that idea is super confusing for you, please feel free to review the chemistry section in the textbook. It wasn't assigned because the assumption is that's part of our prereqs is that you have a basic understanding of chemistry. But anywhere there's a junction like this, there's going to be a carbon. There's a carbon there, there's a carbon there, there's a carbon there, and a carbon there. That's true for the other components here. So anywhere you have a junction like that, there's going to be a carbon in those corners. So all over where you see those points, there's carbon there. And that's because we're carbon-based life forms. We have tons and tons of carbon in our atoms. So going back to the structure of a nucleotide, from the oxygen, we count the carbons clockwise, and we refer to them as primes. So 1 prime, that's this carbon, 2 prime, this carbon, 3 prime, this carbon, 4 prime, this carbon, and then outside of our little person where their hand is, that's the 5th prime carbon. It's not up top because that's where the oxygen is. So we have carbons 1, 2, 3, 4, 5 prime, and then we attach our phosphate group. That's important because that helps us orient our strand of DNA. When we attach another nucleotide, we always attach another nucleotide to the 3 prime end with the phosphate group. So 3 prime, phosphate, 5 prime, 3 prime, phosphate. 5 prime, 3 prime, phosphate. So it helps us organize ourselves. When we look at this strand, we're going from 5 prime to 3 prime. 3 prime is where new nucleotides will be added later. And then this one, note that it's upside down in reference to the first side here. We're going from 5 prime to 3 prime. If we looked at our nucleotides, here's our oxygen on top. Here's our little person with their 2 arms and a little leg. So here we have our 1 prime, 2 prime, 3 prime, 4 prime, 5 prime carbons. And this one is pointing the opposite direction. So that's where we get our vocab word anti-parallel. They're parallel, but they're going in opposite directions. So they are anti-parallel. That's important because whenever we see a 3 prime, we know that the complementary side must be 5 prime, and they are going in opposite directions. This is from 5 to 3, and this is from 5 to 3, opposite direction. Fabulous. So now let's look at the process of DNA replication. There's a whole lot of enzymes involved, and it kind of helps to walk through step by step. So we're going to start up here in this overview of the origin of replication. This is the first piece of DNA that's going to get separated so that the 2 strands are accessible. And in those 2 strands, we're going to have a first primer laid down. So a primer is laid down. A primer is a piece of RNA, not DNA, so there's uracil in it, and it's laid by the enzyme primase. That first primer allows your DNA polymerase 3, which is the main enzyme adding nucleotides, to start adding nucleotides towards the replication fork. The replication fork is where the DNA is splitting, and I want you to imagine you're unzipping it. And so this will continually unzip and unzip and unzip. And as it unzips, this strand can just keep growing toward the replication fork. And because it's continuous, it's called the leading strand. If we go back to our overview here, at the same time, we got a little primer, that piece of RNA. It's done by the enzyme primase. So here's the word primase. Here's a visual of a primer. A primer is a small piece of RNA that allows DNA polymerase 3, who adds the new nucleotides, to attach to your new strand of DNA that it's making. And then that DNA polymerase 3 will continue to add nucleotides toward the replication fork, like a fork in the road, as it continues to unzip and unzip and unzip. It'll keep going toward the replication fork because it can continually add nucleotides to the 3 prime end. And as this opens up, there'll be more and more space for it to continue to go. So the leading strand is easy. 1 primer, DNA polymerase 3 attaches, and it just goes and goes and goes and goes. And you get one on each side of your replication bubble, which is what you call this when there's a replication fork on both sides. And that's great. But what do you do about the other side? Because DNA polymerase can only add nucleotides to the 3 prime end, that means it can only continue going in this direction. So it can't synthesize continually along the 5 and the 3 respectively. And so what has to happen is your primer will add a little piece of RNA with primase that'll tell DNA polymerase 3 where to attach. And it will add new nucleotides to the new strand it's building until it hits the primer there. And then it will stop. And as this is constantly unzipping, you'll get another primer over here, and you'll add DNA until you hit this primer. And you're left with these tiny little chunks. And these tiny little chunks of DNA that are being made by DNA polymerase 3 are referred to as the lagging strand. It literally just takes longer. There's way more primers, there's way more effort, and so it lags. The little tiny chunks are called Okazaki fragments, and those little chunks are awesome, but they're not attached together yet. So now we have to bring in some new enzymes. The first thing that we need to do is we need to get rid of these RNA primers. We can't have RNA in our DNA, and so we use DNA polymerase 1. DNA polymerase 1 will come and it will remove the RNA and it will replace it with DNA. So that's wonderful. But then we have all of these little chunks that are still not attached together. So we need someone to come in and fix these gaps. And the enzyme that does that is called DNA ligase. DNA ligase comes in and it creates a phosphodiester bond between these two nucleotides, and that attaches those little pieces together. So it's really important that it's continuous. As this is happening, we want to note that this is semi-conservative replication. It's semi-conservative because one side of your DNA is original parental DNA, and then as you're making new DNA, one half of your DNA is new nucleotides. So when you're done, half of the DNA is new and half is old. So it's semi-conservative. While we're unraveling the DNA, we have to make sure it doesn't wind up on us. Think about like if you have a rubber band and you twist it and then you pull on it, it gets really super coiled. That's what happens to DNA too. And so we have to use single-stranded binding proteins to hold onto the single strand of DNA. These are single-stranded binding proteins. Otherwise, it'll get all junky and it'll get tangled. And so those will hold those in place so we don't tangle our DNA. And the enzyme that is breaking these hydrogen bonds, allowing the DNA to unzip, is helicase. Helicase, think helix, and then it's destroying the helix. So it's unzipping the DNA. It is breaking the hydrogen bonds between the nucleotides of the two strands. But note that the phosphodiester bonds are not broken. So it just separates like pulling apart, you know, a banana peel or something like that. Wonderful. Let's look at another way to visualize this. Here we have a different drawing. It helps look at more than one schematic. On this drawing, we have an overview again. Up here, we can see our origin of replication. So this is our replication bubble. We have two replication forks. So we have a leading strand and a lagging strand on both sides. Here's your leading strand on the left, and here's your lagging strand on the left. They're going in opposite directions because DNA polymerase III can only add new nucleotides to the 3' end. Think that DNA polymerase III loves the 3. So that's the only side that it can add new nucleotides. Why? That's because the 3' carbon will accept the phosphate of the new nucleotide. That's why we counted the carbons, so we can think about their orientation. You don't want to insert a nucleotide upside down or backwards, so it has to go 3' attaching to the 5' phosphate. So you pop it in right where it goes, and that's why it can only go in one direction. The leading strand will be continuous. It'll add new nucleotides moving toward the replication fork as long as the replication fork is opening. The replication fork is being opened by an enzyme called helicase, which is undoing the hydrogen bonds between the complementary base paired nucleotides of the parental DNA. You also have some other enzymes like topomerase or gyrase that helps hold things together so you prevent supercoiling. Supercoiling is also prevented by single-stranded binding proteins because this single-stranded DNA here is very vulnerable. You want to make sure it doesn't get tangled up or messed with. Note that this says sliding clamp. I'm going to show you an image of what we think this really looks like in a minute. It's pretty cool. I won't ask you any questions about the sliding clamp on exams, but I do want you to know what we think is happening in real life. And so it's pretty beautiful. DNA polymerase III, this is the heavy hitter of DNA reproduction. It's the one that synthesizes the new strand of DNA in our semi-conservative replication. So all of this new stuff here is being stuck together by DNA polymerase III. It takes the nucleotides and it creates the phosphodiester bonds between them. On our lagging strand, which we have a lagging strand on both sides, the right and the left side, the leading and the lagging. On the lagging strand, we have to go away from the replication fork because DNA polymerase can only add to the 3' end. And so we're going to be moving away, which means there's going to be new DNA constantly exposed that needs to be replicated. and we end up making these little short chunks of DNA called Okazaki fragments. Here's Okazaki fragment. The first thing that you need is a primer. Primers are laid down by the enzyme primates. They're composed of RNA. That primer will tell DNA polymerase III where to bind and where to start, and it will continue synthesizing new DNA until it abuts into the next primer. So you end up with these short little fragments of DNA that you need to stick together. But before you can stick them together, you have to remove the RNA primer. The RNA primer is removed by DNA polymerase I. DNA polymerase I will replace all those uracils, and now you have gaps between the two pieces of DNA, and you need to add a phosphodiester bond and get rid of that gap. The enzyme that does that is DNA ligase. So DNA ligase will come and bind the Okazaki fragments together, so you have one nice long strand of DNA, just like you do in the leading strand. So a lot more steps for our lagging strand because DNA polymerase III can only add nucleotides to 3', and so we get these little chunklets of DNA. I mentioned that sliding clamp. Let's look at it. Here is the leading strand synthesis. And sorry, I'm gonna put on my spotlight. Here's the leading strand synthesis. Here is helicase. Helicase is opening the DNA double helix. So this is the parental DNA up here. I've split the DNA. I added a primer. And now once I add a primer, DNA polymerase III will continue adding nucleotides to this leading strand as helicase continues to unzip this. But on our lagging strand over here, we need to make short little chunks of DNA. And as we do, primase adds a primer, then DNA polymerase III will add the new nucleotides. So here's the primer, here's the new nucleotides, and it will continue to do so until it hits this next primer. And it kind of pulls it out and then hooks it back together and then runs through it, and then pulls it out, hooks it, and then runs through it. If you can think like you are trying to pull a hose towards you and you like pull apart and then you organize it, and pull apart and pull and then organize it. So this is the clamp loader. The clamp's attached to DNA. Here are all your single-stranded binding proteins for your single strand, and that allows the DNA to be pulled sideways and worked on and then looped through. And so we don't think it's like all the little enzymes running along the DNA. We think that this clamp loader manipulates the DNA and brings it to one localized spot, and that DNA replication occurs there. You'll be able to see this in one of the animations I'm going to show you at the end. I have two scientist spotlights. The first one is for this husband and wife team, the Okazakis. They published their Okazaki fragment paper in 1968, and it was a huge deal. They didn't term it Okazaki fragments. We named it after them because they're such fabulous scientists. And it's just fun that they were a husband and wife research team. Unfortunately, Rieshi, the husband of the pair, died of Hiroshima-induced leukemia in 1975, and he was only 44. He was 14 when the atomic bomb was dropped on Japan by the US. But Tsuneko is an incredible researcher. She's been active this entire time. She was the first female professor at a major university in Japan back in like the 80s, like 86 or something. So she's incredible. She has some major publications, including helping discover the structure of RNA primers and helping us figure out transcription regulators. So she's just impressive. She's currently 92 and she's still active. So she is a wonderful scientist. And that's where the term Okazaki fragments came from. I also want to highlight the two researchers that won the Nobel Prize in 2009. We have Elizabeth Blackburn and Carol Greer. They discovered the enzyme that helps fix telomeres. So telomeres, we're going to talk about it right now, they are the tips of chromosomes. And it seems that when they shorten, it determines the lifespan of that cell. When they get too short, the cell stops dividing, and we think it's involved in aging. What Elizabeth Blackburn and Carol Graeber did was they studied a tiny little organism that lives in ponds called Tetrahymena, and they discovered that it didn't seem to be aging like at all. Its chromosomes weren't shortening. And so they were wondering how these little critters were fixing this mystery of the shortening telomeres. And they discovered an enzyme. And the enzyme is called telomerase because it works with telomeres. And here's what happens. So when I have my DNA replicating and I get all the way to the end, my continuous strand, my leading strand, can just run right off the end and everything is fine. But for my lagging strand, I get to this end part where I can't add new DNA. And the reason I can't add new DNA is because there's not enough space right here for a primer. And if there's not enough space for a primer, I can't get DNA polymerase 3 to latch on. And that means that this new piece of DNA is going to be that much shorter. And if you think every replication, it shortens, it shortens, it shortens, it shortens. Here we have a karyotype, and we can see on the end in yellow, those are the telomeres that have been stained a different color. So you can see them, they're the protective little tips of the chromosomes. And if those protective little tips are shortened, the chromosomes essentially like fray and start to become mutated and no longer work, and then your cell dies. So what these two scientists discovered was in Tetrahymena, they have this little enzyme called telomerase, and it adds a DNA template, and it allows the DNA to extend itself longer than it should be. And because it's longer than it should be, you can add a primer now, and you can increase the amount of DNA, and the gap that's left over, the part that goes away, would be bonus. And so you're not actually shortening your telomeres. You extend your DNA too long so that when you lose a little bit, you're not shortening the telomeres. A link to this TED Talk. It's from Elizabeth Blackburn. It's The Science of Cells That Never Gets Old. It's really cool. I highly recommend it. And I also have some animations to go through the process of DNA replication, because for me personally, it really helps to see it happen. As you do, think about what enzymes are involved. Think about the difference between the leading strand and the lagging strand. And one of these will show you the really cool clamp loader, which will hopefully make more sense when you see it in action. But it like grabs everything and moves the DNA around so it's localized. To end us, I want to do some practice questions. The first practice question is, which of these is involved in unwinding the DNA double helix before replication? Answer it yourself, and then I'll give you the answer. A, helicase. DNA polymerase can add new nucleotides to what end of an existing DNA strand? 3. DNA 3 adds to the 3' end. Where is DNA polymerase 3? Is it the pink oval, the yellow square, the blue squares, or the green cube? DNA polymerase 3 is the blue squares. What is the point of what number 3, the green cube, is doing? That's going to be primase. And the point is B, DNA polymerase needs a 3' end in order to begin. Hence the primer, because it adds a little bit of RNA, and there's a 3' for DNA polymerase 3 to start adding new nucleotides. Wonderful. I hope you have a great rest of your day. Email me anytime with questions, and I will talk to you soon. Bye.